Today I would like to explain about the mechanism of enzyme action. Enzyme is a catalyst which accelerates the rate of biochemical reaction, or enzyme is active in catalytic action of biochemical reaction. So they act on substrate to form complex after interaction with the enzyme. So enzymes act on the substrate to form a complex after the interaction with the enzyme. The enzyme and substrate. The enzyme and substrate forms a complex at the active site. So here active site or active center of enzyme molecule. So active site, the enzyme has an active site. This active site where the substrate can bind. So such region is calling as an active site or active center of an enzyme molecule. So the binding action makes both enzyme and substrate stable. The interaction between substrate and enzyme may be either ionic bonds, hydrogen bonds, or van der Waals forces. So the interaction between the substrate and enzyme may be either ionic bonds, hydrogen bonds, or van der Waals forces. The active site of enzyme have some special groups such as NH2 amine group, COOH carboxylic group, etc., which binds the substrate to form a intermediate compound that is calling as a enzyme substrate complex. So when the enzyme reacts with the substrate or enzyme substrate binds with enzyme, that form as a intermediate compound that is calling as a enzyme substrate complex. So this reaction releases some energy which raises energy level of the substrate molecules, thus activating substrate molecule, and the phenomenon known as activation energy or energy of activation. So after combining of this enzyme and substrate which releases the energy level of substrate molecule, so which activating the substrate molecule, and that phenomena is calling as a activation of energy or energy of activation. So the mechanism of enzyme action that explained by two theories or two models. One is lock and key model and another one is induced fit model. So let us first know about the lock and key model. So this lock and key model was proposed by German chemist Emil Fisker in 1898. According to him, enzyme have particular shape. So according to him, enzyme have particular shape into which substrate fit exactly, and that refer to as a lock and key hypothesis. So enzyme has an active site. So here the active site of the enzyme rigid. So here substrate act as key whose shape is complementary to lock, and the specific substrate molecule are found to specific region of the enzyme molecule, that is calling as an active site. As I told, active site where the substrate can bind. So the enzyme substrate both have the complementary structure which during complex formation fit to each other like lock and key. So here this enzyme which reacts with the substrate. So here in this enzyme, this is active site where the substrate can bind like this. So enzyme react with substrate and form as a intermediate complex, that is enzyme substrate complex. So the enzyme reacts with the substrate molecule and it form as an intermediate complex. This complex in turn will react to release the enzyme and yield the product of reaction. So lock and key fit between the shape and chemistry of its active site and the shape of the substrate, described as lock and key model. So each enzyme only binds to one substrate. So this is about the lock and key theory or lock and key model. Now let us know about the induced fit model. So here induced fit model was proposed by Daniel Koshland in the year 1958. According to him, enzyme and substrate do not have strictly complementary structure, but enzyme has flexible active site. So in this lock and key, so here enzyme and substrate have strictly complementary structure, but in this induced fit model, enzyme and substrate do not have strictly complementary structure, but enzyme has flexible active site. So here enzyme and their active site are flexible structure. Look at this figure. Here this is enzyme and this one is the substrate. So here enzyme and their active site. So active site are flexible structure that mold themselves into a precise shape of substrate, which enables the enzyme to perform its catalytic function most effectively. This model can be compared with the type of hand and glove. So this is the example for induced fit model. So hand and glove. So the same glove can be fit in hand of many persons. The glove adjusts itself in the hand of those persons who wear it. The conformational changes in the enzyme are induced by the substrate. So here the changes are induced by the substrate. This active site can mold by the substrate. So here formation of enzyme substrate complex. Then it releases enzyme and their products. So this theory suggests that an enzyme undergoes a changes in its structure when it reacts with a substrate molecule to form an activated complex, and the enzyme regain to its original structure after the reaction. So here mechanism of enzyme action can be explained by these four cycle or catalytic cycle. So first one, the substrate binds to active site of the enzyme. So like this. So here substrate first binds the active site of the enzyme. Then second, binding substrate induces the enzyme to alter its shape. So here the binding substrate induces the enzyme which alter its shape. Then third, active site of the enzyme breaks the chemical bonds of the substrate and new enzyme product complex is formed. So here in this active site of the enzyme, it breaks the chemical bonds of the substrate and form a new complex product. The enzyme releases the products of the reaction. So here enzyme release the product of the reaction and free enzyme is ready to bind another molecule of the substrate. So free enzyme, it ready to bind the another molecule of substrate and run through the catalytic cycle once again.